All right, everybody, we're going to pick up and finish up our meiosis notes that we started previously. And so, kind of just a quick recap, we went through meiosis one, which is kind of the first stage of meiosis. We call that the reduction division because we took the amount of chromosomes that each cell had and we reduced it by half. So, for example, if a chromosome originally or a cell originally started with 4 chromosomes, it would end up with 2 at the end of stage 1 of meiosis. So, now we're going to talk about meiosis 2. And so we left off, we just talked about telophase 1, which is where essentially those cells started to pinch off, those 2 cells, and then cytokinesis, where they split. Now, remember, during telophase, just like in meiosis, those chromosomes are going to start to get uncondensed. They're going to kind of go back to that kind of stringy mess instead. And then we end up going to prophase 2. So prophase 2 is similar to mitosis in the fact that you have the chromosomes recondense. So you would see them back in those X-shaped structures. You have the nucleus has reformed, and you'll still see your centrioles there. So let's switch back and look at our fill-in sheet quickly. Okay, and if we look here, it actually doesn't have us draw any specific pictures for prophase 2. And the reason being is because it looks so similar to telophase 1. Essentially, we have those chromosomes back in the X-shaped structure, so we're not even showing them changing really, even though they probably would decondense and then recondense. But instead, we would see that the nucleus would have fully reformed. But we're not going to take the time to draw it just because it does look so similar to what we've already drawn in the previous pictures. All right, so let's keep moving. Let's go back to our PowerPoint here. Okay, metaphase 2. So like mitosis, we now have those chromosomes. They're going to line up in the center of the cell or the middle of the cell, which is how you remember metaphase, think middle. So all of those chromosomes then in each individual cell, again, they're lining up in the middle and they're essentially attached to those spindle fibers that are attached to those centrioles. Let's go back and draw our picture. So I'm going to go ahead and draw in my centrioles here. And then I'll draw in my spindle fibers in just a moment, but I'm going to go ahead and draw my chromosomes and draw them in the middle. Now it doesn't matter, so I'm going to look at these 3 first for this cell here, just going to go straight down. It doesn't really matter which orientation I put them. I could put this big one over here, and it's possible that maybe this whole chromosome flipped over. So I'm going to just draw it here just for the sake of kind of keeping it similar. Here's its little green end there. Okay, I'm going to go ahead and draw the green one next to it. And then I have the little one. Okay, then I'm going to go ahead and draw in my spindle fibers attaching to each of those chromosomes. Sometimes you'll see it kind of with like some extra fibers in there. Okay, there's our first cell. Let's go ahead and let's do the second one. Okay, now again, it doesn't matter if I flipped it around and I either put this end here or I could put it on this side because it's possible that the chromosome would have flipped over. Not a big deal either way, as long as I'm drawing that same chromosome there in the middle. Okay, so there are my chromosomes. Let's draw in those spindle fibers. Okay, so there are my pictures for metaphase 2. And let's go ahead and let's go back and write in our key events over here in the box. Okay, so for metaphase 2, we're going to say that individual chromosomes lined up at the center of the cell. Okay, metaphase 2, and again, it looks just like what it would look like in mitosis. The only difference is those chromosomes have some little pieces from their homologous pair where they did that crossing over process. All right, let's switch back to the PowerPoint. And let's look at our next phase, which is anaphase 2. Okay, anaphase 2 is now where those sister chromatids get separated from each other. Those spindle fibers pull on that X-shaped chromosome and pull it apart so that one half of it goes to one side and the other half goes to the other side. So again, we're pulling apart those sister chromatids, and those chromatids are essentially moving to the opposite ends of that cell. This looks just like it does in mitosis. Again, the only difference being that now some of these chromosomes have little parts from that homologous pair from that crossing over process. Switch back to our pictures. Let's go ahead and let's draw those in. And again, now I have to keep it consistent. So if I'm looking at this individual cell here, then this half of this chromosome is going to go this way. This half is going to go that way. That half is going to go up that way, and so on and so forth. So let's draw that in here. Sometimes I find it easier to draw the chromosomes first. Okay, and a lot of times we kind of just flip the direction, but if I'm looking at this right here, it's essentially this shape with a little bit of green on one end. Okay, now I'm looking right here. It's just all completely green, that half of it. And then on this one, completely pink then. So I want to just kind of make sure you can see this, because sometimes I hover over it and I'm not sure you can actually see what I'm trying to show you. So I'm just going to use this color here. So this one right here is essentially pointing to this piece. Okay, this part right here is this piece, and this part right here is that piece right there. So just so you're understanding where I'm kind of going with this, I'm going to go ahead and get rid of all of that here. Chromosome. So now I'm looking at this half right there, okay, the half that went pulled this way. Oops. All right, so let's switch colors again. Okay, and on this one, the pink half just looks like that. Okay, on the next one, it's green with the little bit of pink on one side of it. And then this one is pink with, oops, a little bit of green on that side. So go ahead and draw in my spindle fibers then, showing that these are attached and they're getting pulled, which is why they're separating. Okay, so again, those X's have essentially just been pulled apart, and now those sister chromatids are going to opposite ends of that cell. All right, let's do the other one. We're going to do the same thing here. And again, I have to keep it consistent. So I'm looking at this part right here. Okay, it's going to look like that. On this next one, the top half of that X shape, or that sister chromatid, is pink with a little bit of green. Then the last one is green with a little bit of pink on it. Okay, on the bottom, we're going to do the same thing. Oops, sorry, I'm going to draw it actually going the other direction just to keep it the same. So green, a little bit of pink. Okay, the next one's just all pink. And then the last one is just all green. Draw in my spindle fibers. Again, pulling those, that's what's separating those sister chromatids. All right, so let's go ahead and let's switch. Let's write in our key events of this phase. And that is that sister chromatids separate and move to opposite sides of the cell. Okay, let's switch back and let's look at telophase 2 and cytokinesis. Okay, now that we've separated those sister chromatids and they've moved towards the opposite end, we're going to start to get the distinction between those cells. So they'll start to pinch off in telophase, and then in cytokinesis, they will eventually split, and you end up with 4 haploid cells. They're haploid because they have half of the information than what they started with, and each of them will be genetically different. Okay, so let's go back and draw our pictures. Okay, and just for the sake of you tracking where I'm kind of putting all of these, so I'm going to put these 3 right here in this cell, put these 3 in this cell. I'm going to put these top 3, I'm going to put them in this one, and these bottom 3 in this cell. So basically what would happen is this would start to pinch off right here, start to pinch off, and then eventually it would completely separate, and so that you would have those 2 distinct cells. So this top portion right here being this top cell, and this bottom portion being a distinct cell. So let's go ahead and let's draw them. And we're going to show that those sister chromatids kind of straighten out. So in this cell, I'm going to have kind of a longer red one that has a little bit of a green end on it. Okay, then there's a kind of a medium-ish green one, and then kind of a smaller pink one. Okay, and then what's going to happen is we're going to get that cell membrane is going to reform. So you can either draw it as a dashed line, because that's what it would be in telophase, but eventually by the end of cytokinesis, it would be completely reformed. So I'm going to just go ahead and draw it all together intact, because we're kind of showing these 2 phases combined. All right, let's do the next one. So this was, again, that top one. Now let's do the bottom one. Okay, there would be a kind of a longer red one. Here's this green one that has a little bit of a pink end to it. And then the small pink one. Oops, sorry. With a little bit of green on it. And again, we're going to draw our nucleus around, because it starts to appear in telophase, and then by the time cytokinesis is finished, we would have it completely surrounding those chromosomes. Okay, next cells. A long green one for the top one. And I'm just going to put this little short one over here already, since I have the green. Okay, there's that one. Okay, and notice I now have 4 cells, and they each have 3 chromosomes, whereas the cells that we originally started with had 6. If you go back and look at your picture, it would have had 6 X-shaped chromosomes. And then remember, even though up here they're still X-shaped, these are still 3 chromosomes, because remember, we count them by the centromere. So here are the centromeres in the middle. There's 3 centromeres, but when I split these, remember, the chromatids still have their own centromere. So this is 3 and this is 3. So we went from 3 to 3. So we stayed the same amount here. We just essentially split apart those chromatids. All right, but notice again, they are all completely different. So we have 4 completely different cells, which is pretty awesome. So let's write our key events in. Okay, 4 haploid cells are produced and each cell is genetically different. All right, so that's it for our pictures. So let's go ahead and let's finish up our last couple slides here. Okay, so this just kind of gives you an overview. It's not showing you all of those each individual steps, but remember in interphase, we have the sister chromatids, they duplicate. So now, again, we went from 4 chromosomes. I still have 4, but they're in the shape of they have the 2 sister chromatids per chromosome. Okay, in meiosis 1, we call that the reduction division. So those homologous pairs, they line up in the middle, and then we split apart those buddies, those homologous pairs. So we end up with 2 chromosomes in each cell. So we reduced it here. We went from 4 to 2. Okay, and then in meiosis 2, it looks really similar to mitosis. These chromosomes would line up in the center, and then we would pull apart the sister chromatids, and 2 would go each way. So I have 2 chromosomes in this cell, but I still end up with 2 chromosomes in each of these cells, but they are haploid because these cells have 2 when our very original cell had 4. And again, notice that each of these is different than the other. Here's kind of another picture, just to kind of put things in perspective. So your mom has 46 chromosomes, dad has 46 chromosomes, but he essentially got one from, your mom got one from her mom, one from her dad. So she is kind of a unique combination, so does your dad. And then essentially you're getting crossing over between those, and then they're lining up. And so you're getting a really unique combination that's between your mom and dad's chromosomes, and then essentially what they got from their parents as well. So that's kind of just showing you that kind of flow of genetic information. All right, and I have kind of a quick animation to show you, and then we'll be finished after that.
Before meiosis begins, in the preceding interphase, DNA replication takes place. At this point, the chromosomes are not condensed and are not visible under a light microscope. However, we depict them in a condensed manner so that you can see that the cell replicates each chromosome to form 2 chromatids. Meiosis consists of 2 cell divisions, meiosis 1 and 2. As the diploid cell enters the first phase of meiosis 1, called prophase 1, its chromatin condenses into discrete chromosomes. The cell has 2 centrosomes, which form the mitotic spindle as the centrosomes migrate to opposite poles of the cell. This diploid cell contains homologous chromosomes, which pair up later in prophase 1. 1 chromosome in each pair is maternally derived, indicated by blue, and 1 is paternally derived, indicated by red. The pairing is called synapsis. At this time as well, the nuclear envelope breaks down. Late in prophase 1, the homologous chromosomes exchange genetic material, as indicated by crossing over points called chiasmata. Recombinant chromatids consist of both maternally and paternally derived DNA. In this phase, microtubules attach to the chromosomes, directing them to the equatorial plate. At metaphase 1, homologous chromosomes have lined up on the equatorial plate in a pairwise fashion, with 1 homolog on either side of the plate. Note that the chromosomes assort independently. For example, the maternal chromosomes align randomly, not necessarily on the same side of the plate. During anaphase 1, chromosomes from each pair move to opposite poles of the cell. The centromeres of the chromosomes do not divide, so each chromosome still consists of 2 sister chromatids, which now may not be genetically identical due to crossing over. In some species, telophase 1 occurs, a stage in which chromosomes decondense, nuclear membranes reform, and the cytoplasm divides in a process called cytokinesis. A short interphase period called interkinesis may then follow telophase 1. Note that DNA replication does not take place during interkinesis. During the second half of meiosis, in prophase 2, the chromatin again condenses into discrete chromosomes. There are now only a haploid number of chromosomes per cell. Each chromosome consists of 2 chromatids joined together by a centromere. At metaphase 2, the chromosomes have lined up on the equatorial plate. Microtubules from opposite poles attach to each sister chromatid of a chromosome. During anaphase 2, the centromeres divide, and both chromatids become independent chromosomes and move to opposite poles of the cell. During telophase 2, chromosomes again decondense and nuclear membranes reform. Depending on the species, cytokinesis may occur.